So let's talk about the concept of epistasis. And these are from Greek words, which essentially means standing upon. This is a situation where one phenotype that an organism displays is dependent on another phenotype or another step in that organism. This is all about genetic pathways and genetic interactions between genes or proteins. And one of the first groups that worked on this process worked with model organisms and their synthesis of necessary amino acids for growth. And what these scientists did was that they found mutants of this organism that couldn't grow in the absence of the essential amino acid arginine. And to get to the root of this problem, they needed to understand the biosynthetic pathway that organisms use to develop or produce their arginine that they use to grow. And that involves a compound called ornithine, I'm abbreviating here ORN, being converted into a compound called citrulline, which is then converted into arginine. And there's a precursor molecule that is turned into ornithine. And each of these steps is controlled by a different enzyme. So there are proteins in the cells that take the precursor molecule, and enzyme X turns it into ornithine, and enzyme Y turns ornithine into citrulline, enzyme Z turns citrulline into arginine, and the organism has created arginine for it to grow and produce proteins that require that amino acid. Now, these scientists discovered a variety of different mutants that were not able to grow unless they gave those mutants the amino acid arginine in their medium. So they knew these mutants couldn't produce arginine. They knew they were mutants somewhere in this pathway. Either gene X, gene Y, or gene Z must be mutant and not producing the enzyme that's needed to convert that into ornithine, ornithine into citrulline, citrulline into arginine. And they called these mutants Arg1, Arg2, and Arg3 for arginine growth deficient. And this is what they observed by doing some studies where they wanted to know which compounds, when provided to these different mutants, 3 different mutants of the same organism, if you put in their media these different precursors, would the organisms be able to grow or not? None of these mutants, Arg1, Arg2, or Arg3, were able to grow just in the presence of the precursor molecule. And again, that's what tells us that somewhere downstream of this step, these mutants are defective. They're unable to convert some precursor into arginine. So here's where they started seeing differences, and they worked out the order of genes in this pathway, which is one of the goals of the study of epistasis. They found that Arg1 mutants could grow if you gave them ornithine. So without ornithine, unable to grow. But if you put in the media these organisms were growing in a little bit of ornithine, then those organisms were able to grow. The same was true if you gave them citrulline, and the same was true if you gave them arginine. And as you might expect, if the problem with these mutants is they're unable to grow in the presence of arginine, if you give any of the 3 of them arginine, they're able to grow. You've given them what they're trying to produce, and they're defective at producing, so they're fine. All 3 can grow in the presence of arginine. What they discovered with the Arg2 mutant is that unlike Arg1, if you gave them a little bit of ornithine, they were still unable to grow. And the same was true for Arg3 mutants. But Arg2 mutants could grow if you gave them citrulline, but Arg3 mutants were still defective at that process of growth. So Arg3 were the neediest mutants. They could only grow if you actually gave them the thing that they were trying to produce to be able to grow, the arginine amino acid itself. And so what this suggests is it tells us which of these genes controls each of these steps. That is, Arg1 can't grow without this precursor, but if you give it ornithine, it can grow. And that suggests that the defect that's problematic in the ARG1 mutant is the conversion somewhere of the precursor molecule to ornithine. That suggests that ARG1, the gene, encodes whatever enzyme it is that converts the precursor to ornithine. Once ARG1 mutants get ornithine, their ARG2 and ARG3 genes are fine, and so they can finish the process of converting ornithine to citrulline to arginine, and they can grow. ARG2 mutants are defective not necessarily in the step of converting the precursor to ornithine, but here is the difference. Unable to grow in ornithine, but once you give them citrulline, they're fine. So that mutation that is present in the ARG2 mutant is a mutation in the ARG2 gene, which must be the gene that's necessary to convert ornithine into citrulline. And likewise, the problem with the ARG3 mutants is that they're unable to grow in the precursor molecule, ornithine, or citrulline, because even if you give them any of those compounds, if you don't have a functional ARG3 gene that converts citrulline to arginine, you could give those organisms all of the precursor molecules you want. They'll convert the precursor to ornithine, they'll convert the ornithine to citrulline, but all of that citrulline just piles up because their enzyme can't do anything with the citrulline to turn it into arginine. So this again is the concept of epistasis, is that the ARG3 mutants, it doesn't matter what precursor you give them, they're all defective, and those genes then are epistatic to this final gene that controls the last step in the conversion of citrulline to arginine, a necessary component for growth in these organisms.